So we are going to look at the questions now. As you can see, IGCSE Biology 0610, that is chapter 4, Biological Molecules, paper 2 exam questions, that is for extended, which is multiple choice from the year 2021 to 2025. So we are going to answer the questions. All right, so this is the first question that we are going to answer, and for this one is coming from paper 2, variant 1, May, June 2025. So the question was on question 5, a DNA molecule contains pair of bases. Yes, we know. What is the correct combination of pair base? What is the correct combination of pair of bases? I said C bond with what? G. Then T bond with A. Okay, it can be AT or it can be TA. It doesn't really matter. Can be G, C or C, G. So long as you know that C bond with G, A bond with T. So which one is correct? A cannot bond with G, C cannot bond with A, G cannot bond with T, so the correct is D. So T and A. What is the test for proteins? So question 6, the same year. What is the test for protein? So name of the test. So the name of the test is Bulate. So we don't care about Benedict and also Benedict there. So it's Bulate. Does it require heating? No. I said only reducing sugars or Benedict requires heating. Then what is the color? Blue to purple. So the answer here is C. Look at how the question comes. Then now let us look at this one, paper 2, variant 3, May, June, that is 2025. In a DNA molecule, bonds between pair of bases hold two strands together. In a DNA molecule, bond between pairs of bases hold the strands together. High temperature can break these bonds to form two single strands. So when they are heated, the bonds can break. The base pair C and G is held together more strongly. You know that C and G is a triple bond. Held together more strongly than A and T. A and T is a double bond. So you expect this one to be strong. But that is not the question. The thing is that two DNA molecules containing the same number of bases were studied. In a DNA molecule 1, 20% bases are T. Now, do you know what it means 20% are T? So look at this. We are saying T pairs with what? Pairs with A. So where you find T, you find also A. If we are saying this is 20%, it means that also this is 20%. So they pair in equal. So 20%, 20%. So out of the total percentage, we already have 40%. So 40% is just for A and T, which means G and C, this one, they share equally. So which means what remains out of 100 is 60. So which means this is 30% and this is 30%, making it 60%. And this gives us 100%. I guess you get that. Now, what is the question? Which DNA molecule would require the lowest temperature to break into single strands? Which one would require lowest temperature to break into single strand? Look at this. DNA molecule 1. Is it DNA molecule 1? DNA molecule 1, say, contain 20% of T, which means it has 30% or more of what? Of G, which are very difficult to break. So this one cannot be. Then DNA molecule 1, because they are more G than they are saying lowest. So this one is different. The more they are, the pair of G, the more heat is required. Now see, DNA molecule 2, because there are more A bases. Do we have more A bases? Which one will require? So DNA A has more of? No. It's supposed to be DNA 2. Look at this. So for 1, we've calculated, is the one that we've calculated. So for 1, if it's 20% for this, which means G is 30%. So this one is hard to break because we have more of this. But for this second one, if it's 20% of C, which is same as 20% of G. So 20% of this, which means it has A of 30%. So more of the A are easy. So in other words, DNA 2, because there are more of A bases present. So this is the answer. I guess you've gotten something here. Then let us look at this question. So this is paper 2, variant 2, February, March 2025. A section of a chromosome contains 4,500 bases. A section of a chromosome, so a chromosome is just where the DNA is coiled. So contain 4,500 bases. There are 1,400 G bases. Now, these are bases, not pairs. There's a difference between 1,400 pairs, which means pairs it will be C, to G, but not pairs. So, which means this is one pair, but this is a base, which means just just G. So there are how many of these? Okay. Then they are saying how many A bases are there. Now, out of the total 4,500, G are these. It means that you also have the same number of C, 1,450. So the total number of these C and G giving us, that is 0, 0. We have this as 9, then I think is 2,900. So it gives us 2,900 for these two. Now we need to find the base of A. So we are going to say 4,500 minus 2,900 is giving me 1,000. So 1,600. Now this 1,600 is for both A and D, because we just want for A, but you know that the pair is 80. So just like we did here, if one is 1,400, which means also the other one is 1,400. Now here we want for A, then we have this for the two bases. So which means we divide this one by 2, and you are going to find 800. So the answer is 800, and I guess you've gotten a thing here. Which chemical element is found in protein but not in carbohydrates? So remember, component of protein, carbohydrate, and fats, CO, but in proteins you have N, which is the nitrogen. So only in protein is nitrogen. Let us look at this one, which is paper 2, variant 1, October, November 2024. Which statement about biological molecule is correct? DNA molecule contain pairs of bases. A pairs with G. No. A pairs with T. So this is wrong. The second one, fat acids and glycerol molecule can be joined to form a protein. No, they form fats. Glucose molecule can be joined to form glucose molecules can be joined together to make cellulose and glycogen. Yes, yes. Starch molecule are made of maltose. Maltase, no, not maltase. If it was maltose, yes, maltase is an enzyme. So the answer here is, you see, glucose molecules, remember among the polysaccharides that I talked of, cellulose was one of them. So cellulose, glycogen, starch are polysaccharides made up of glucose. So the answer is C. Then we look at this question, that is paper 2, variant 2, October, November 2024. Which base in a DNA molecule always pair with G? So the one that pairs with G is C. By now, I'm sure you are familiar with this. Let us look at this one. So this is paper 2, variant 3, October, November 2024. Which food testing reagent shows a positive result when it turns from blue to purple? Which food testing reagent shows the positive result when it turns from blue to purple? Look at how clever questions are asked. So here is butyllate. You know that you are testing for proteins. Then let us look at this one. The diagram shows the stick end of the DNA of plasmid that has been cut by a restriction enzyme. The unpaired bases are labeled. So these are unpaired bases. Now, what is the correct sequence of missing base? So the missing base down here. Now, you need to know that C pairs with G, this one with T, this one with G. We have G there, so this is C, this is A, this is C. So GT, CA, C, GT. So we are looking for GT, GT, CA, C. So this is the answer. Question 8. Test was performed on the food substance. A positive result was shown by the color change to blue-black. Remember, blue-black is starch. Which solution was used to test? So they've not asked what kind of or what was being tested, but they've asked us of a solution. As I said, you need to make sure that you know the solution. So in that case in this case is iodine solution. It's the one that is used to test for starch. Let us look at this one. That is paper 2, variant 2, May, June 2024. The diagram shows the bases in the section of the DNA from one strand only. So, from one strand only, this. What is the base sequence of the complementary strand? So, which means the opposite one. So, it's going to be T, G, A, G, T, C, A, G. So do we have TGAA? So we want the one that is starting with T. TC, no. So TGAA, so already is this one. All right, the diagram shows 4 molecules. C represent carbon, H represent hydrogen, O represent oxygen, and N represent nitrogen. Which structure is an amino acid? So we are going to skim through or look through these diagrams and look at the one that has the nitrogen, because amino acid is related to protein. So we are looking for the one that has nitrogen, and it is this one. So B is the amino acid. This is paper 2, variant 2, 2024, that is February, March. Which row shows the element that make up a carbohydrate molecule? So carbohydrate made up of carbon, hydrogen, oxygen. So where there's carbon should be yes, where there's hydrogen should be yes, where there's nitrogen no, then oxygen yes. So this is the answer. Question 11, large molecules are made from smaller ones. Which row names the large molecule and the small molecule it is made from? You remember that I said such questions you find them. So for me to explain, which means I have seen quite a number of questions. So large molecule cellulose made from smaller molecule glucose, yes. Large molecule fat made from glycogen, no. Glycogen, fat acid, no. Starch, amino acid, no. So the answer is A. This is paper 2, variant 1, October-November 2023. What is the color change shown by Benedict solution when heated with reducing sugar? So reducing sugars are present. So Benedict solution is blue. It changes to red or yellow or green or brick red. So blue to purple, no. Blue to red, yes. Brown to blue-black, this is iodine. Red to yellow, no. So the answer is B. Paper 2, variant 2, May, June 2023. DNA contain pair of bases. Which pair shows correct combination? So A cannot bond with C. C and G yes. G and A no. T and G no. So the answer is B. Paper 2, variant 2, October-November 2023. The first question was, which row identify the chemical element in protein? So here we are talking of protein. So protein contain carbon, contain hydrogen, oxygen, and nitrogen. So throughout should be ticked, and that is A. The table shows large biological molecules and small molecules. So large biological molecule. We have cellulose, oils, glycogen, protein. Now, small molecules. So we are looking for a corresponding small molecules. So the first one is the answer, that is cellulose, glucose. Oil, amino acid, no. Fat acid and glycerol. Glycogen and glucose, yes. Then protein and ethanol, no. So it is 1 and 3. Which row correctly pair large molecule with smaller molecule used to make them? So 1 and 3, which is B. This is paper 2, variant 3, October-November 2023. Which row shows the chemical element contained in fats? So fats contain the same as carbohydrate, carbon, hydrogen, oxygen. So shouldn't be nitrogen. So nitrogen should not be ticked. So we are targeting this one or this one. Now the fact that this one is not ticked is not the answer. So the answer is A. Glycogen is a large molecule made from smaller molecules. Which smaller molecule glycogen is made of? So it's made from glucose. The diagram below shows part of the protein. So this is the protein, we've been told, protein molecules. Now, protein molecules is made up of smaller units. These smaller units called amino acids. So these are amino acids. What does X represent? So represent amino acid. Question 8. That is, structure of a DNA involves two strands coiled together to form a double helix. Which pairing of bases between the strand is correct? So C cannot bond with T, C cannot bond with A, T can bond, so it's B. That is paper 2, 2023, May, June, that is 2023. Paper 2, variant 3, May, June 2023. The table shows the result of a test carried out on the fruit or on the fruit. So this test was carried out on the fruit. So Benedict solution, you know that Benedict is for sugar, so positive. Bullet for protein, positive. Ethanol for fats, which is ethanol emulsion, negative. So iodine solution, negative. So this food or this fruit had no fats and no starch, but had proteins and sugar. So what did the fruit contain? So protein and reducing sugar. Question 8, describe the structure of the DNA. So how is the structure of the DNA? Each strand coils, or each strand contain chemicals called fat acids. No, each strand contains sequence of amino acid. Each strand contains mRNA. Two strands coil to form a double helix. So two strands coil to form a double helix. But is this also the possible? What makes B the wrong answer? So which one is the answer here? So put up a debate on this part. Let us look at this paper 2, variant 2, February-March 2023. Which substance is used to test for vitamin? So what is used to test for vitamin is DCPIP. Question 8, in the DNA, what is the correct base pairing? So TT, no. TC, no. TG, no. So it's TA. The diagram shows a length of a DNA. So this one is paper 2, variant 1, October-November 2022. What is the sequence of bases in an parallel or in an unlabeled strand of DNA? Starting from the top of a diagram. So starting from the top of a diagram, unlabeled part is this one. So what does A pair with? T. What does T pair with? A. What does G pair with? C. C, G. Then this one, T. Then this one, G. So it will be TA. So the one that starts with TA. So not this one, not this one, not this one. So TA, CG, TG. Yeah, so the answer is B. This is paper 2, variant 2, October-November 2022. DNA is a molecule consisting of two linked strands. Part of a single strand of a DNA molecule has the sequence this. So part of it has a sequence of this. What is the sequence of the second strand? So the second strand needs to correspond with this. So on the first part, it will be T, then here G, then here G, then here C, then here A, then here C, then T, T. So it should be TGG, CAA, CTT. So the one that starts with the T. So TG, G, that is the AACTT. So the answer is C. This is paper 2, variant 3, October, November 2022. Which statement about the structure of a DNA is correct? DNA contains 6 types of bases? No. DNA contains 3 strands coiled to form a helix? No. 2 strands. DNA is made up of a chain of amino acids. DNA has 2 strands which close link between the base pairs. So this is correct. Question 9. A doctor tested the urine of 4 patients for both, so look at this, glucose and protein. The color of the urine sample after testing are shown in the table. So color of testing for Bullet, so Bullet blue, blue. So the one that remained blue, then here color for testing for Benedict. It cannot turn, brick red. Then this other one remained blue. Now what is the question? Which patient urine contained both protein and glucose? So protein and glucose. So we need purple on the protein, then brick red on this other part. So in other words, it's C. This is paper 2, variant 1, May, June 2022. What are the smallest units or basic units of a starch and glycogen? So starch and glycogen, they are made up of glucose. So we should find glucose, glucose. So the answer is T. In the length of a DNA, 20% of the bases were T. So T, 20%. Now T pairs with A. It means that also A is 20% because these need to be in equal proportion. What is the percentage of G? So G and C, we are going to share the percentage that remains. So this is 40% out of 100. So what remains is 60%. Now 60 divided 2, so which means this is 30%. And also this is the 30%. So the answer is 30%. This is paper 2, variant 2, October, November 2022. What are the smallest basic unit of starch and glycogen? So this one is the one that we are from answering. So the answer is D. Then here is a question on question 8. A chromosome was analyzed and found to have 6,000. So 6,205. So let us answer question 8. So for question 8, a chromosome was analyzed and found to have 6,125 base or pairs of bases. Now, if these are pair of bases, you know that pair of bases is a pair, this and that. So there are 2. So which means this one, 1,025 multiplied by 2. Now, if you multiply, what I want is to find the total number of bases. Okay, so these are pairs, but to find the total number of pairs, the bases you multiply by 2. So 6,125, that is 6,125 multiplied by 2. So it's giving me 12,200, please, 12,200, that is the 12 to 250, like this, 12,250. This is the total bases. Now we are told just for G, there were 2,345. Now, to multiply by 2, because these are equal or in equal proportion, if G were 2,345, which means also the corresponding C was this same number. So multiply this number 2,345 multiplied by 2 is giving me, out of these 12,250, 4,600 were bases of C and G. Now, if you subtract this from 12,000, what remains are the bases of A and T. So minus, that is 12,050. I'm finding 7,560. Now, this is total number of A and T. So divide or share that number by 2. Okay, so divide 2 and it's giving me 3,708. So the answer is B. I'm sure you've gotten the explanation here. Then question 9, which type of molecule are enzymes. So enzymes are proteins. So this is the part that we are yet to look at enzymes, but enzymes are proteins. Let us look at paper 2, variant 3, May, June 2022. What are the smallest basic unit of starch and glycogen? So this question has repeated even here. Question 8, DNA is made up of 1, is made up of dash strand coiled together. So it's made up of double strand or 2 strands coiled together. The strand contain chemicals called bases. So it contains chemicals called bases. These chemicals form dash. Okay, so cross link. So we are looking for 2, then bases, then let's see, it's made up of 2 strands, then of bases, not 3. 2 strands of bases cross link. So the answer is B. Let us look at paper 2, variant 2, May, June 2022. Which row is correct for a positive test for vitamins? So vitamins, color of the food test reagent. So DCPIP is blue. So you're going for blue, not colorless. It turns colorless upon adding the vitamins. Heated, no. So no heating. So already know this one has yes heating, so we are going for this one. Then final color of the test reagent, blue. No, it becomes colorless. Okay? So blue, no, colorless. So this is the answer. All right, we've left out one question. In which molecule are close links formed between bases, DNA, to form a double helix? This is paper 2, variant 1, October, November 2021. The bases on one of the strand of a DNA molecule have the sequence. What is the corresponding sequence for the strand? So I'll leave this one to you. I've answered quite a number of questions on this. And of course, I guess this part is very simple now. Then let us look at this one. That is paper 2, variant 1, May, June 2021. This one should be the continuation. Yeah, it's the continuation of what we've not answered. What element is found in protein but not in carbohydrates? Nitrogen. The diagram below shows the short section of a DNA strand, that is the strand of a or single strand of a DNA. Which strand of a DNA will combine with this strand to form part of a double helix? So TGGA. TGGA. So TGGA, which is this one. Then paper 2, variant 2, May, June 2021. Which element is found in protein but not in carbohydrate? This one we've answered already, nitrogen. Found in protein but not in carbohydrate. Okay. The sequence of bases present on one strand of a DNA molecule, this one. So these questions I've answered quite a number of them. Which sequence shows the base present on the opposite? So on the opposite, you're going to have TAA, CC. So TAA, CC. So this is paper 2, variant 3, May, June 2021. Which element is found in protein but not in carbohydrate? Same question. The DNA sample was tested to identify its bases. A DNA sample was tested to identify its bases. 35% of the bases in the DNA was G. So G was 35. Now, if G is 35%, also it means that C, which is corresponding to G, is 35%. Now, these two make up what is 70%. So which means what remains 30% is shared A and T. Now, what is the question? Which row shows the percentage of other bases? So we have A, C, T. So this is 35, 15, 15, because we share this into 2. So the answer is B. This is paper 2, variant 2, that is February, March 2021. The bases on one strand of the DNA molecule have the sequence C, A, G, C, T. So which means the corresponding is G, T, C, G, A. Okay, so GTCGA. So this is the answer. And this brings us to the end of the presentation. So please, if you are still here up to this point, do not forget to subscribe and also share with others. This is the detailed information that you need for your exams. As you can see, these questions were taken from the exams and of course the latest exams. So as you write in 2026, that is 2027, 2028, this is what you need. Otherwise, see you in the next one.